You guys, this is a really fun one. This is a biggie, right? Methylation, everybody's so excited about methylation. I'm excited about methylation, and I have to thank Ben Lynch as the spearhead for me. That's where it all began, and my learning began in, I don't know, maybe 2007. But needless to say, what an integral part of our health, and I'm really excited to talk about it with you, and I hope to give you some new information and not just talk about the pathways, but underlying clinical aspects of presentation that we want to consider when we're dealing with methylation defects. So our learning objectives are going to be define methylation, the fundamental interrelationships that we're seeing, understand the key enzymes and their function, determine the genetic analysis and what's going to be appropriate for the patient based on their results, learn how to interpret the panels, and then identify key labs that can be utilized in conjunction. So really that part will mostly be covered in our lab matches made in heaven, but I'll touch on that in this lecture as well. So what is methylation and why does it matter? Methylation is actually super, when you really think about it, it's pretty darn simple, right? We have methyl groups of carbon and 3 hydrogens. It's very, very simple. But the movement of that methyl group is incredibly complex and important to DNA methylation, silencing retrovirulent elements, regulating tissue-specific gene expression, genomic imprinting, and like Corey said, X chromosome inactivation. Oh, sorry. There we go. So here's that little enzyme, this little molecule. And what's fun is that uracil, thymine, is just a methylated version of uracil. So when we're talking about RNA versus DNA, that's a fun little tidbit. So methylation in terms of single nucleotide polymorphisms is our focus today. We're really going to just be looking at one key aspect of methylation in the body. But this really gives us an opportunity to analyze metabolomics in a totally different light. I'm sure most people have seen this study. I think I first learned about this from Amy Yasko a long time ago, but it's a really fun study on basically epigenetics and long-term health outcomes, and taking these agouti mice and having them, pregnant mice, fed a folate-rich diet and a folate-deficient diet, and what the outcomes were in their offspring. And the diet that was low in folate, we found the yellow, yucky coat, more obesity, and prone to disease, as compared to the diets rich in methyl groups. We have these healthy, brown, shiny coat mice with healthier outcomes, and they're not prone to disease. So basically, we already know methylation is going to affect homocysteine levels. We'll look at the pathways on the next slide and throughout this lecture. Epigenetic regulation is influenced by methylation very specifically, a large role in cancer and aging. And then methylation and glutathione regulation are important overall. So this is just a general methylation map. Not every pathway on this map actually has to do with the transfer of methyl groups, but the integration of these cycles is what we consider the methylation map. I'm going to focus on each section and break it down so that we can talk about what we're testing, why we're testing certain SNPs versus others, the cofactors involved, foods that can support them, deficiencies that you're going to see in your patient associated with deficiencies in the cofactors, so that we're not just looking at, okay, what's the enzyme and what does it do, but what are you going to see in front of you that can help you work with this beyond just your lab tests. And let's start with the folate cycle here. And forgive me because my slides overlap a little bit between the folate and the methionine cycle, so you'll see that along with me here. Here's our folate cycle, and we see our lovely happy little mitochondria here. And this is where we're getting dietary folate is coming on in, and there are enzymes all along this pathway, but the big enzyme that we're looking at is over here, where we go through all these steps to get folate into 5,10-methylene tetrahydrofolate. And that's what we need in the methionine cycle. And the relationship between, MTR here, which is methionine synthase, So it can also be considered MS depending on whose presentations you're looking at. But that's also linking the folate cycle and the methionine cycle. Oh, those got a little bit wonky. All right, so functions that require efficient methylene tetrahydrofolate reductase activity. You probably already know most of these. Purine synthesis, pyrimidine synthesis, obviously homocysteine metabolism, methionine production, nervous system function. There are over 200 pathways that require the transfer of methyl groups. Some of the really big ones are the production of creatine and choline. So we'll get a little bit more into depth on those as well. Big cofactors that we want to consider when we're looking at the methylation cycle include riboflavin as FAD, and this is also going to involve thyroid function, right? T4, one of the very clear roles that we know about free T4 or thyroxine is that it has a relationship with the production of FAD. So this is one of those places where it's a holistic picture. You can't just look at one tiny piece of information. If we don't have optimal thyroid function, you could be supplementing riboflavin, but that doesn't mean you're producing FAD effectively. NADPH is required, zinc, we already know the amino acids, and then SNPs that are indirectly linked include MTR. MTRR, this is methionine synthase reductase, that has to do with the recycling of cobalamin. BHMT, betaine homocysteine methyltransferase, which is essentially the shortcut across the methionine cycle. We'll talk about that in a minute. And SHMT, which we don't have a great deal of research on. This is one of those places where you'll get those particular programs where you can input your genetic information, and it might tell you the SHMT outcome, but research-wise, we really don't have anything solid to support whether SHMT has associations with particular pathologies. Interactions, overlap, and support. So weak MTHFR is going to strain the other enzymes. So if we're struggling, if we were consuming a folic acid-rich diet and preventing the uptake of folate, as well as dealing with a slow MTHFR enzyme, that's going to strain our MTR, our CBS, and our BHMT pathways. So that can also cause secondary deficiencies in the cofactors required for them. B12, choline, betaine anhydrous. These are all things that are going to be affected by that. If we're taking a bunch of folate and only looking at MTHFR and not MTR and MTRR, we can end up with methylcobalamin pooling. And that's going to slow down the pathway. So we really need to take a look at all of this together. We're going to see indirect effects of the NOS or urea cycle, the biopterin cycle is actually directly related, and then transsulfuration. But I wanted to point out some of the side effects with these nutrient deficiencies because I think in the United States at least, even when you're trying to eat a pretty darn healthy diet, as Corey was discussing earlier, our soil is depleted. We're inhaling toxins. The burden on our body is a lot higher, and we're going to run through these pathways more rapidly. So when you want to look at your patient, you really want to look at not only what are their single nucleotide polymorphisms, but what are the signs of deficiency in front of you? And I don't necessarily need to run through these with you, but this is just for you to go home and say, hey, that patient has chelosis, and they've got, some polymorphisms, and they've got some hoarseness in the throat. Maybe we're dealing with a riboflavin deficiency, and I should be emphasizing some dairy in their diet, assuming that they can manage dairy effectively, or fish or eggs. There's also supplementation you can use. My goal is generally to minimize too much supplementation. I really want to work on gut healing, and I hope that we all do, because I've been on 15 supplements a day, 3 times a day in my life, and that is a challenge to maintain. Niacin deficiency can be, here are some foods that support niacin in the body. These are from the Oregon State Linus Pauling Institute website. Great resource, so much information on nutrients that you can utilize. I really appreciate the information accessible. And then zinc deficiency. This one's hard. I work with children in particular, and really oysters are going to be the biggie in terms of getting zinc into the digestive tract. So I often find that it's necessary to supplement with zinc, but just an FYI, you can utilize any of these foods as a starting point, or really a point after you're healing the gastrointestinal tract. So some of the research out there these days, I wanted to just touch on it, and then I'll talk about some of the pathologies too. But this particular study, I don't have a highlighted part here, but so the finding of this research provide evidence that homocysteine and S-adenyl-homocysteine, which are biomarkers of one-carbon metabolism, and they're influenced by genetic and nutritional factors and their interactions. So diet is a huge aspect of things, and I guess I need to have an argument with Corey's dad because there is solid research out there. This one's just talking about the increased risk of ischemic stroke. I think we're all pretty familiar with the elevated homocysteine being related to cardiovascular disease. And then this one down here is talking about the interaction of MTHFR, MTR, and RR, elevating hyperhomocystinemia risk. And those are all acting on that same methylation pathway, which I'll get onto the methionine cycle in a moment. Some of the indirect interactions with MTHFR could be COMT and MAO and ESR1. This is one of my favorite studies, not one that we could use in terms of choosing the single nucleotide polymorphisms that we test because it's a small study on Polish women, but it's a nice one in terms of the explanation on how these interact. So if that's something you're ever interested in or you work with menopausal women, I think this is a really interesting study to look at. But we do know that MTHFR is associated with Alzheimer's. It's associated with interactions with COMT, so depression, schizophrenia, bipolar, hormonal imbalances, and then MAOA is linked to depression as well as ESR1. And then I wanted to put a note on BHMT here, because when we talk about the methionine cycle, we'll delve a little bit more into betaine homocysteine methyltransferase, but the research isn't as strong as we would like it, so it's not something we test, but it's really an important enzyme to take into account when you're looking at the methionine cycle. Because when you look at this, okay, so this particular map is the opposite of the one I was showing you earlier. So we've got the folate cycle here, and we've got the 5-methylene tetrahydrofolate coming in. Here's methionine synthase, MTR. And if we are not functioning appropriately here, right, we don't have enough folate, the enzyme's super slow, we're going to have sluggish action here, and we're going to build up our homocysteine. So this is our body's sort of backup mechanism, and we're going to use betaine to create DMG and convert the homocysteine into methionine from a sort of side street, if you will. And I like to incorporate, you can see it here, that choline is required to create that betaine anhydrous, which pushes the pathway here. And then, as you work on looking at single nucleotide polymorphisms in combination with other labs, you can utilize things like, sarcosine, well, that's going to come up in an organic acid test. So you can sort of make these connections based on looking at these different labs in combination. But needless to say, this is interesting to consider, and I think it's something that we should definitely be aware of, although at this point the testing is not there. And do know that, choline can, if you supplement choline, it can push that pathway and affect neurotransmitter production down the line, causing mood issues. But here are some of the studies. They tend to be smaller or more specific, but there's that interaction between MTHFR and BHMT. There's some relationship with liver genotypes and phenotypes, and then some really interesting stuff on non-alcoholic fatty liver disease So I've also seen clinically that not only using betaine anhydrous, but also using phosphatidylcholine can bring liver enzymes down. I had a 30-year-old female, and they were ready to biopsy her liver, and we started some phosphatidylcholine based on testing. And 30 years, I mean, this has been going on probably 15 or 20 years. They've known her liver enzymes were elevated, and that was all it took. Phosphatidylcholine, we made some diet interventions, but finally her enzymes came into balance and we avoided a biopsy. Some of the other research here is a relationship to preeclampsia. Male infertility is a big one. And then there's this association with these polymorphisms and ALL. Okay. Another associated pathology we already talked about was Alzheimer's in the graceful aging section. And now, okay, so we already talked about the methionine cycle when I told you about the shortcut. But let's delve a little bit more into depth here. The methionine cycle is where we have methylfolate coming in to this cycle. It interacts with MTR, methionine synthase, and then there's sort of an off cycle here where we see that cobalamin converted to methylcobalamin, and based on the polymorphisms there, you can make an assessment whether methylcobalamin, adenosylcobalamin, or hydroxycobalamin are going to be more efficient for your patient. Because if you give a patient who has a high-functioning MTRR but some dysfunction in MTR methylcobalamin, that might push things a little bit too much, and they might actually do better with something like a hydroxycobalamin, and even cyanocobalamin. Some of the cofactors here, well, we need the folate there. We're going to need some zinc here. And let's see what's on my next slide. Oh, so this is a little bit redundant, but we're just continuing to talk about how this pathway is. That folate's coming in. We need adequate B12 and B12 recycling to convert that homocysteine to methionine. So then over here, we can get our S-adenosine methionine to transfer our methyl donors. And these are the methyl groups that we need in order to feed that one carbon metabolism all over the body, create our neurotransmitters, work on those detox pathways. And I think it's fun to just point out, methionine is essentially just methyl homocysteine. Okay, so cofactors for this particular pathway we sort of already discussed, but cobalamin, and depending on the patient's MTR and MTRR status, you're going to determine whether they need more methylcobalamin or hydroxy, and you can sort of look at the interactions between the SNPs to determine that. Methylfolate, zinc, choline, and magnesium. And I will just mention again, choline, you want to be careful because so many of our one carbon donors are responsible for the production of choline, that if you add phosphatidylcholine to someone's diet and it pushes the pathways that create our neurotransmitters, we can sometimes find our patients having mood disturbances. So you really want to be really conscious. I really am a strong proponent of starting low and slow and individualized in terms of the supplements that we're using. Homocysteine and methionine, that's repetitive. And then the SNPs involved here we'll talk about a little bit more. Interactions, overlap, and support again. So weak single nucleotide polymorphisms in the methionine cycle are going to result in reduced cell proliferation, reduced protein synthesis, RNA synthesis and transamination are going to be affected. And I didn't get into it quite as much, but folinic acid can also be really beneficial for certain patients rather than the methyl tetrahydrofolate, because based on their clinical presentation, I work with a lot of children with Down syndrome, so DNA repair is incredibly important in making sure that we have those purines and pyrimidines, and folinic acid plays a very big role in that. So it's not just the methylfolate that you want to use, you might want to use a combination. Folinic acid and calcium folinate are the same, FYI. Signs of cofactor deficiencies. So this one's really not signs because it's risks, but high homocysteine is going to be a sign of potential methylfolate deficiency, and these are just increased risks you might have. Choline deficiency could be muscle damage, muscle pain, myopathy, and abnormal deposition of fat in the liver. Zinc we already talked about. Magnesium, you're going to see that there is potentially reduced vitamin D and calcium homeostasis, increased risk of cardiovascular disease, but really more, you're going to see a patient who potentially has bruxism. They could have muscle cramps, restless legs, or fatigue. Magnesium is just required in over 300 biochemical pathways. Most Americans are pretty darn deficient. So I'll talk a little bit about the forms of magnesium, but almost everyone needs it at some point or another. And then methylcobalamin, we can get stomach inflammation, anemia you'll see on lab tests, potentially neurological disorders. That's going to be a lot further down the line. More commonly, you're going to see fatigue. And if you're working with fertility patients and they're dealing with recurring miscarriages, methylcobalamin and methylfolate and choline, honestly, are all going to be big players in that. Vitamin B12 sourced foods. We've got a lot of great foods that are high in vitamin B12. So as long as your patient isn't a vegan or a vegetarian, you're in a good spot. I like to support my patients' dietary choices personally. B12 is in some mushrooms and maybe in brewer's yeast, but I do really encourage most vegetarians and vegans to either eat eggs or take in some supplementation. And it takes about 5 years to deplete your stores, right? So a vegetarian is going to be deficient after 5 years of cutting out all meats and eggs and fish. Choline food sources, egg yolks specifically, but the whole egg, beef, scallops, as well as organ meat. And then folate, Corey already gave us a really lovely example of that. Magnesium, okay, magnesium's high in brown rice, but we've got to be so careful about too much brown rice because rice can have higher arsenic, and then we can contribute to thiamine deficiencies, as arsenic requires B1 to detoxify it. And then white rice specifically, you want to try and avoid in excess in any patient because of thiamine deficiency and arsenic levels. Spinach is great, nuts are great. And then we want to look at the different forms of magnesium for our patients. Not every person needs the same kind. And like I said, I work with kiddos with Down syndrome. So many deal with constipation. So I often will use a combination of magnesium citrate. Magnesium citrate works pretty well as an osmotic laxative. It doesn't cross the gastrointestinal lining very effectively. So although you're going to get some benefits with magnesium deficiency, you're also going to get gastrointestinal benefits. So you'll get a little bit of calming. Magnesium glycinate and bisglycinate is a nice one for patients who need something calming. This is going to cross the gut barrier more effectively. Glycine is one of the precursors to the production of glutathione. So if we have a patient with oxidative stress, this is going to be a lovely option. You always want to, I like to look at organic acids or amino acid levels in order to get a sense of where my patient is, but I do find that this is a lovely option for many people. There's some really new interesting research on magnesium threonate crossing the blood-brain barrier. So when we're talking about patients who are anxious, maybe have some neuroinflammation, and maybe mitochondrial dysfunction, magnesium threonate is a really nice option. And then magnesium taurate is going to be more calming. Taurin's a bile acid precursor. So if we're working with digestive dysfunction and potentially poor bile production, that's a nice option too. Okay, this is a little bit of, so we're not talking about MAT1 today, but this is within the methionine cycle as well. And this is actually a smaller study that it's 288 subjects, right? So these are the kinds of studies that we can look at to see how it's going to affect the methionine cycle. but it's not something that we're testing at this point because there's just not enough out there. Did I pass something? Oh, here we go. Methionine synthase polymorphism and cancer risk. So here's a nice meta-analysis. So we're not just talking about hyperhomocysteinemia and the type of B12 the patient needs, but when you put together the pathways, and we'll review that methylation map together, but we're going to potentially increase cancer risk on many, many levels with some of these polymorphisms. But you don't want to tell a patient who's going to look at a SNP as a death sentence, but that's for us practitioners to know. So much information on pregnancy, fertility, and neural tube defects with folate and B12. And then this is just a little picture of some of the polymorphisms that we associate with Alzheimer's. So this one's talking about MTHFR, methionine synthase reductase, cystathionine metasynthase, and serine homocysteine methyltransferase as all being involved. But like I said, the studies are not huge. This is a 2001 older study, smaller population, so we don't look at that particular polymorphism, but we sure as heck hope that the research keeps expanding so that we can utilize these SNPs for more information. Genetic variants involved in 1-carbon metabolism, just more influence on methylene tetrahydrofolate reductase and homocysteine levels. And this is one, obviously, that I look at a lot, folate metabolism and maternal risk for Down syndrome. So MTHFR, MTR, those are all pretty big risk factors. Okay, so far we covered where our folate's coming in and how it's passing into the methionine cycle, and this is where all our big methyl groups are being donated. And as we move down, this is where our homocysteine produced. So if we don't have good movement here, we're going to get an increase in homocysteine. But, excuse me, I was talking to Joan earlier because she has a patient who is homozygous for C677T. And that is a significant reduction in the efficiency of the MTHFR enzyme. So you would assume that patient's going to have high homocysteine. But we don't look at one SNP because that's not the reality. This patient's homocysteine was fine. That could mean this BHMT pathway is working well. Maybe we need to supplement some betaine anhydrous and choline to support this pathway, as well as support methylene tetrahydrofolate with some folate and some B12 in here. Or maybe the CBS is upregulated. And the research on that is definitely not as strong as some others, but we do look at it, and we do see that CBS can shift this pathway, especially in Down syndrome. There's some significant research on CBS.
I had one patient who's had fluctuated between methionine and homocysteine, and she went to the several kind of holistic practitioners before when she came. And she came with the blood work was done, and during different periods of her life, like sometimes months apart. And she's been using different nutritional supplements for treatment, right? And there were fluctuations between, there was a period when homocysteine was high, and then methionine was high homocysteine. So there's some in that cycle, but she was trapped in that cycle, no matter what.
Did you get to the bottom of it?
Yeah, I did, but the patient did because she was completely she was
compliant.
She was like something.
Well, and that is one of the hardest things I think in the work we do.
I got her betaine very small amount, and then she improved. But then she started using a lot of it.
Right.
And it was just very interesting case. But she's had few genetic variations there,
and that's why it's great to look at them in combination.
Yeah, but the interesting thing that in a dynamic of time, that's what I try to say to you, that when you watch those patients, maybe that person who's at normal homocysteine at that point, she wouldn't have it
3 months
at 2 weeks or whatever after.
Yeah, certainly, and that's another really good point. So we do want to pay attention to serial testing of homocysteine and pay attention to what's going on, right? Because these are just, lab work is a moment in time, right? We've got to look at our patient, not just the paper, not just the lab results. Okay, so transsulfuration is a pathway down where we're taking our homocysteine and breaking it down into cystathionine. I love this pathway. CBS is expressed on the 23rd chromosome, so it comes up in my daily life with all my patients. But this is a very B6 dependent pathway. Cysteine is required for the production of glutathione. You'll see that over here. But if this pathway is dysfunctional, if we're in a high inflammatory state, if we're weak in our cofactors, we can end up with elevations in ammonia. We can end up with elevations in oxidative stress and sulfates. So it's awesome because we can use things like B6 or P5P, the active form. With ammonia, we can use NAG or N-acetylglucosamine to help support breaking that down, or look further at the urea cycle because ammonia is coming in here. With sulfation, we want to consider molybdenum. And then taurine, this is why we look at labs in combination, right? Because we don't know exactly what part of the pathway is being pushed. But when we have enzyme dysfunction here, any one of these can be affected, and we want to support our patient based on their presentation and the combination of pathways. SUOX is a nice addition to the transsulfuration pathway, but yet again, that's one that we're really struggling with this solid research that we can all agree upon. Okay, so this is just a closer look. We see that up here in the methionine cycle, this is where we're getting our 1 carbon donation, and down here is where we're getting our glutathione synthesis. And anytime, I mean, we all know glutathione, right? But you have to also consider reduced versus oxidized glutathione in your patients. So cofactors you may need to support transsulfuration include B6 and magnesium. I don't know why I didn't put molybdenum is on there as well. N-acetylglucosamine is a little bit off the transsulfuration cycle, but like I said, that could support ammonia breakdown. We can look at all these different amino acids. So if you do test amino acids and you see discrepancies in the values, so I often will see all the amino acids low or all of them leaning to the right. But if you are seeing just elevations in these particular ones or some of these, you can definitely look back and say, okay, what's going on with the methionine cycle? What's going on with the transsulfuration cycle? What, which single nucleotide polymorphisms do I need to consider? Cystathionine beta synthase enzyme hyperfunction is going to potentially reduce homocysteine. So just because you have that MTHFR, you can't assume that there's going to be a high homocysteine level. It could be working more slowly, so you could have even higher homocysteine. And then we already talked about the different things that could be affected. But the signs of deficiencies, so pyridoxine, pyridoxine 5-phosphate or pyridoxine HCl, whatever you choose to utilize, this is essential for it's essential to over 100 enzymes, right? So it's a cofactor in so many pathways. We know that microcytic anemia could be related to it, irritability or depression, sores and ulcerations in the mouth. And a lot of people will get recurring sores in the mouth. And I feel like that's a symptom we often are like, oh, maybe you had a virus, or, just sort of overlook it, but we really do need to hone in on these things. And then neuropathy, like I discussed, we could have too much or too little B6 contributing to neuropathy. Magnesium we talked about already, but because it's required by ATP synthesis and we need ATP, this is sort of a double whammy. And then some of the things we need for ATP production. Foods that are high in B6, so fish, poultry, nuts. This, like I said, this Linus Pauling Institute website's a really good resource for looking into the different nutrient deficiencies and how they present and what you can do for them. In terms of research with CBS and the transsulfuration cycle, this one we see that CBS is linked to homocysteine levels based on the folic acid concentrations. And then this one here is talking about cystathionine beta synthase and its relationship to oxidative stress. Logical when you look at the biochemistry, but we can't make an assumption. This is the same thing. Oh, here we go. Here's CBS and non-alcoholic fatty liver disease and the relationship there. So now I'm just talking just a tiny bit about liver detoxification pathways because we were talking about transsulfuration and glutathione. But this is really for you to review and how the glutathione conjugation in the liver is important for so many different aspects of detoxification. Okay, the biopterin cycle is really fun because it's sort of like an unheard of pathway. We don't typically supplement biopterin, although you can. In the US, it's not something we can access readily, and I don't necessarily think it's a good idea to supplement tetrahydrobiopterin. But the biopterin cycle has a lot of an effect on the urea cycle. It's going to affect the folate cycle, so indirectly we're going to see these pathways affected as well. And then as you see here, tryptophan and tyrosine coming in from your diet, coming in from your supplements are going to be affected by the availability of tetrahydrobiopterin, which is then going to affect serotonin and dopamine levels, and it's going to interact with the polymorphisms in MAOA and how we have tyrosine coming in and using tetrahydrobiopterin here, and tryptophan using tetrahydrobiopterin to create serotonin, and then this is where we're making a big connection with catecholamine O-methyltransferase and monoamine oxidase. And this is a little bit of a picture where we have inflammation and oxidative stress. We're going to have more of a toll on our tetrahydrobiopterin. So we need that tetrahydrobiopterin to get our phenylalanine to tyrosine, or tyrosine to L-DOPA. We know that L-DOPA then goes to dopamine, tryptophan 5-HTP to serotonin to melatonin, and arginine to nitric oxide. I mean, these are all reliant on biopterin. So this is an integral piece of information that we really want to consider. And when we're taxing the system, we end up with BH2 or dihydrobiopterin, which is just not as readily available. Then if you have issues with the methylation cycle in terms of the methionine cycle and carbon donation, we can't recycle back to biopterin. So that's why rather than using biopterin as a supplement, if you can optimize your patient's methylation in terms of folic acid versus folate and the methylcobalamin or hydroxycobalamin, you're going to optimize biopterin that way. Cofactors in the biopterin cycle include B6, methylfolate or folinic acid is going to be effective, iron, niacinamide, and then we already talked about the amino acids and some of the other single nucleotide polymorphisms. I jumped the gun there. So interference with that biopterin cycle I sort of broke down, but neurotransmitter balance will be affected. We're going to have a slower breakdown of nitric oxide, or a slower creation of nitric oxide. And then these pathways can create excitotoxic quinolinic acid in the brain, and I'll talk about that in a minute. So signs of cofactor deficiencies: iron deficiency, fatigue, cramping, bruising, poor cognitive development, microcytic anemia, and then amino acid deficiencies can manifest in a million ways. I like to show this image because this is where biopterin is really important, right, in these conversion of tryptophan here. And when we have tryptophan in a really inflamed situation, this pathway here is affected by biopterin. We don't have the movement towards serotonin. Instead, we end up converting into kynurenine. And kynurenine and quinolinic acid are two important acids to look at in terms of neuroinflammation. When the relationship between quinolinic acid and kynurenic acid is off, or we have an elevated level of quinolinic acid, we're going to have more excitotoxicity in the brain and more oxidative stress, and a shift in our NMDA receptors, which can result in all sorts of neurological pathologies and presentations. So back to cofactors, iron, you always want to look at TMP or SS6, or I like to. And then the difference between heme iron and non-heme iron. Always consider this when you're working with vegetarians and vegans. Some food sources of amino acids. I do like to be really conscious of utilizing phenylalanine, tyrosine, and tryptophan before I work on gut function. If you ever are interested in organic acid testing, we know that there can be an elevation in quinolinic acid and kynurenic acid when we're in an inflammatory state and there's Clostridia species. So when you take phenylalanine, tyrosine, and tryptophan, Clostridia species can utilize those to increase the quinolinic acid levels. So this is where we just keep going back to, you really got to work on your patient's overall health before loading them up with too many supplements, and you really need to look at the whole picture. Okay, so another study on methylation and neurotransmitters and antioxidants linked to depression. Methylene tetrahydrofolate reductase can be related to monoamine neurotransmitter biosynthesis. Tetrahydrobiopterin regulates monoamine neurotransmission. We already looked at the biochemistry. It makes sense, but you need the research to back it up these days. And that's reasonable. Biopterin relates to endothelial nitric oxide synthase, and we'll look at that in the urea cycle right here. So our urea cycle sort of gloms onto the methylation map by the folate cycle feeding the biopterin cycle, which then interacts with nitric oxide synthase here. And endothelial nitric oxide synthase we know is really responsible for vasodilation. But as you can see here, if we have dysfunction in nitric oxide synthase, or if we have dysfunction in the biopterin cycle, our production of citrulline and nitric oxide can be reduced, and we can end up with peroxynitrate and superoxide. So that's going to lead to neuronal damage and microglial activation, oxidative inflammatory disease. And the reality is most people have a significant amount of oxidative stress. So how do we utilize this information in order to optimize treatment for our patient? When we can pinpoint these little idiosyncrasies in the enzymes and the pathway function, we can look at the clinical picture, work on the pathways, optimizing or bypassing these pathways in order to support the patient. Cofactors here, B6, pyruvate, biopterin, tetrahydrobiopterin specifically, magnesium and glycine. And then we already talked about the amino acids and the single nucleotide polymorphisms affected. Dysfunction in the urea cycle can cause all sorts of problems, right? In my kiddos, I see flapping a lot, hand flapping, and that can often be related to higher ammonia levels, so that could lead back to this cycle here. Or looking at their genetic testing, I can see, oh, well, wait, there's nothing going on at NOS, but we've got some issues with CBS, and is that where we're dealing with elevations in ammonia? So needless to say, we want to consider ammonia levels, and then when we're looking at any patient with neurological symptoms, right, ammonia can cause some significant neurological disorders. I already talked about the peroxynitrate and such. Reduced ornithine, we've got a lot of research on ornithine and arginine, which are in the urea cycle, affecting sleep, the circadian rhythm cycles, as well as growth. So I work with kids who don't grow particularly well often, and so these are things that I really take into account. Signs of cofactor deficiencies, when we're talking about the cofactors in the urea cycle, most of them are redundant because we've talked about a lot of them, but you could see dermatitis, cheilitis, glossitis, neuropathies, and then we're going to see things like fatigue and lack of endurance. So some of the research on nitric oxide synthase and NOS polymorphisms is a relationship to an increase in the progression of diabetic neuropathy, or nephropathy, excuse me. Then we see insulin sensitivity and adiposity being affected by nitric oxide synthase polymorphisms. And then this shift, so it's this is the polymorphism that we look at Kashi Labs. Then we see that that's related to LDL oxidation and can affect, and the patient, based on their polymorphism, is going to respond differently to smoothies, essentially. So I thought that was really fun and interesting. And people with the genotype, their vasodilation after a vegetable and fruit puree-based drink is going to vary based on that NOS polymorphism. So I thought that was a really fun one, and it also links nutrition back in. Granted, that was not a research study we're basing our testing on. Nitric oxide synthase has a significant relationship to metabolic syndrome and cardiovascular disease, increased hypertension. That's sort of just logic, but we also like to look at what the research says. Okay, so we will talk about this more in the lab tests made in heaven, but monitoring methylation, what can you do beyond looking at SNPs? And I think we already talked about this a little bit, but serum testing of B12 and methylmalonic acid, or if you have the ability, sometimes intracellular testing with something like SpectraCell can be a really nice way to look at B12 levels, because honestly, in clinical settings, I don't find that this is always the most reliable information, and like we know, serum testing is a moment in time. We can also look at folate levels and folate levels in combination with homocysteine. And when you look at folate levels, you want to talk to your lab and say, well, what the heck are you measuring, right? Are they measuring folic acid? Are they measuring methylfolate? Are they measuring calcium, folinate? What are we looking at? Because that is a big factor too. You don't want your MTHFR compound heterozygous patients' folate levels to look great, but it's because they've got a boatload of folic acid in their system. That's no good. Whole blood homohistamine. So this is not a research-based. I mean, it is research-based, but it's not meta-analysis type of information. But whole blood histamine elevations are thought to be linked to poor methylation. So that's something you can look at. Doctor's data does a SAMI-SAW ratio, so that's going to give you an idea of the ratio of methylation that's happening. I'm sure there are other labs as well. Oxidized glutathione versus reduced glutathione. And then, more importantly, look at your patient. Watch for their change in emotions, sleep, blood pressure, digestion. Talk to them about side effects from these supplements, because, gosh, we're so casual with supplements. Probably we're not so casual, but people are very casual with supplements, and we can have significant negative side effects. And in terms of patient retention, it's important to discuss these things. So dietary lifestyle factors for DNA methylation. Corey already talked about this, but there is research. There's solid research that dietary and lifestyle exposures on DNA methylation can be modified by the genetic variants, as well as environmental carcinogens, infection. I mean, these are things that this is what we get to do. We get to delve into these underlying causations for pathologies in our patients that are overlooked in many conventional medical settings. This is just another one pushing the importance of nutrition and epigenetics and optimal nutrition. And I would venture to say gastrointestinal function is going to improve epigenetic expression. This is an interesting on DNA methylation in children with asthma. So supporting methylation could potentially improve asthma outcomes. And you want to support detoxification in your patient with any of these polymorphisms, right? We know that detox is going to be affected on so many different levels, and toxicity is everywhere, right? where our food is toxic, unfortunately, our water is toxic. This is one of those arguments that you often have to have with patients. Sometimes you might have to have it with yourself. Our homes, there's some really interesting research on environmental toxicity if you live within a mile of a highway, which is like, if you live in a city, you live within a mile of a highway. So taking into account all the different factors that are going to affect our health, the water that we're consuming. Not only are we worried about, what heavy metals, right? Poor Flint, Michigan still has lead in their water. But what about the drugs that we find? I know the salmon, well, let me see, was it the salmon or the shellfish in Seattle are found to have high levels of different medications, because where is it going when you flush it down the toilet or you pee it out? It's going into other food that we're going to be eating. So we really need to be conscious of these things and support all of the emunctories, right? We want to support respiration, sweating, elimination, all the different things that I discussed earlier. Minimizing stress, and I feel like we can't overlook this in our patients, but taking that time to actually teach them how to breathe. People don't know how to breathe. I forget how to breathe all the time, but we just live in a very fast-paced world, and we all breathe up in our chest. So if you can take the time to slow the body down, we know stress is going to affect all of these pathways. So if we can just slow our bodies down and be present, that can really help. So whether you want to support meditation, or if you want to suggest whatever it is that puts your patient in the present moment, you can consider adaptogens to help manage stress that way, exercise, and then don't be so busy making a living that you forget to live. Because money drives so much in today's society and life that we want to remind our patients that there's more to it. Food hygiene, I'm sure you guys know about, and we already talked about, digestive elimination and healing the gastrointestinal tract, exercising daily, incorporating saunas if you can. That's not one that I use in my pediatric patients, but they're, in other countries they often will use some sauna time in kiddos, or you can use hydrotherapy. Watch for nutrient side effects and interactions. We already sort of talked about this too, but you want to start low and go slow. You don't want to give 5 supplements all at once, because how the heck do you know what's going on? And there are obviously exceptions to that rule, but man, a probiotic can cause changes in behavior or just changes in digestion. So if you're giving a probiotic and you're giving methylfolate and you're giving choline, how do you know? How do you know? So low and slow. Provide your patients with treatments to manage their side effects. So if we are doing a gut treatment and you feel like a person's dealing with die-off, then what do they do? Can we use vitamin C to get a clearing bowel movement, or molybdenum to support sulfation, or charcoal to soak up the endotoxin? And most importantly, we're treating the patient. We're not treating the single nucleotide polymorphism. So that's why we always look at more than just the SNPs. We look at the patient, and then we look at correlative labs, or labs that can complement genetic